Can somebody please confirm you can hear me?
Yes.
Are you hearing me well?
Yep.
Good. All right. Let me ask, are there any questions? No questions, so I'm going to move on. Let me share my screen. There, you should be able to see my screen now. All right, today is September 29th. And we should be starting, or we've already started chapter 2. We should be working on chapter 2 today. Remember that there is a practice quiz. It is actually due Wednesday, but I think I've set that up so that you can actually take that a day late. I see a number of emails. I have not looked at my emails today. If you haven't taken the practice quiz, let me know and I'll open it up for you. Because it was shut down yesterday. If anyone's having trouble with Respondus Lockdown browser, don't contact me, contact the computer help desk. Any questions about any of that? Let me remind you that we do have some extra credit assignments. And one is you can send me a picture of your face and I'll give you 3 or 4 extra credit points. I think one of them is already done. You're supposed to give me your study schedule. And then there's the extra credit or EC study 2. Form a study group, get together with another student or students and quiz each other, study some material that you will be tested on in this class. It can be either the lecture or the lab. All it needs is to be is material that I may quiz you on. And then one person in the group needs to submit to the extra credit study group folder, which we'll have a link over here or a link down here for you guys to upload it. I've got a link to SpeedGrade it. And what you need to do is tell me what you studied, when you studied. So give your full name and the full names of everyone in your group. It does need to be the full name because, especially with the first name, there's more than one student who may have that name. So tell me when you studied, and it's usually the day you submit your document, what you studied and how long you studied. You do need to study for at least a half an hour to get credit. You may not work on an assignment together, then submit the assignment for credit as well as extra credit, meaning you're supposed to be using this extra studies credit group to help each other study and then get ready for the quizzes. So if you do work on an assignment, I'm not going to penalize you for doing that, but you're not going to get extra credit. Okay? Meaning you're already going to get credit for that assignment. I'm not going to give you extra credit. If everything is submitted correctly, everyone in the group will get 1 extra credit point per session, and you may do 1 session for each week up until 10 weeks. Any question about any of that? All right. Note that there is a worksheet for lecture 2, which is due this Saturday. And generally, you only answer a question or two. And this one will be dealing with chemistry. It looks like a large number of people have already submitted answers. We finished lecture 1, so let me go ahead and open the PowerPoint. If that isn't open, oops, yeah, that's right, chapters 1 and 10, then just send me a reminder to open it, because I forgot until now, actually.
Just to clarify, we can have that PowerPoint.
I'm sorry?
Just to clarify, we can have that PowerPoint. It's
Yeah,
on, it should be on Canvas.
we're done with the lecture.
Where is that
It should be in Canvas. I just showed you. At the bottom of week 1 lecture, right there. All right. I think that's it. So let's go ahead and begin the lesson. Guess I'll leave that up for the lab. All right, we had talked about what a polar molecule was and mentioned that water is a polar molecule, having polar covalent bonds within the water molecule, holding the hydrogen to the oxygen. And then it has hydrogen bonds between water molecules. That is why, let me see if I got some water. Of course, I don't know if you can see that. That is why water will take the shape of the container it is in, why it holds together and will change the shape to whatever container it's in. It's because of the hydrogen bonds holding the water molecules together, and they break and they reform, giving us liquid water. If ever the hydrogen bonds were to never break, we wouldn't have liquid water. What would we have? Come on, somebody's got to know.
Wouldn't
So
it be
the
ice?
hydrogen bonds between the water molecules do not break. What do we call this?
Isn't
Not
it
liquid
ice?
water. We call it. All right, think about that. And then if the bonds were to totally break, we don't call it liquid water either, meaning the hydrogen bonds were to totally break. We call it steam. If there's no hydrogen bonds between the water molecules, we have steam. All right, we need to talk about water is a good solvent. Water is a good solvent because all polar molecules can dissolve in water. Ions can dissolve in water as well. And in the loose wording, you can call an ion a polar molecule. It's really not a polar molecule. An ion is a molecule that has a charge, like sodium chloride, is a charged substance. Let me blow this up a little bit. Where the sodium ion has a positive 1 charge, and it's held together in salt to the chloride, which has a negative charge. And the attraction is simply the positive charge is attracted to the negative charge, making the salt crystal. However, if you put water around the salt crystal, the water will orient itself so that the negative, partially negative oxygen will move towards, where's my mouse? Will move towards the positively charged chloride. And this is sodium, and that's a positive 1. This is a partial negative charge. And then you get several water molecules surrounding the sodium, and then it can lift sodium out of the sodium chloride crystal and then make it dissolve in the water. And that's what we have as we have salt water, where the negative, partially negative oxygen is oriented to the positive 1 of the sodium. And then the partially positive hydrogen of water is oriented to the negatively charged chloride ion. Any question about any of that? And this attraction between the positive sodium ion and the negative chloride ion in salt, we call that an ionic bond, because there is an electromagnetic attraction between these two atoms. And then the salt just arranges, looking at it here, the positive +1 positive sodium, my mouse keeps dying, will be surrounded by 4 different chloride ions, and then we get a crystalline structure of the sodium chloride. But the point is that both a polar molecule like glucose can dissolve in water. And what happens with glucose is that the negative oxygen will be attracted to the partially positively charged hydrogen in glucose. And that's why sugar or glucose can dissolve in water. And that's true for all polar molecules. And then ionic molecules take on this form as well. And that makes water a good solvent because it can dissolve all polar molecules and all ionic molecules. However, water does not dissolve nonpolar molecules, such as lipids, like when you mix vinegar and oil together in your salad dressing and then you shake it up, the oil will separate from the vinegar and the oil will raise to the top and the vinegar will raise to the bottom or lower to the bottom. Any question about any of that? So polar molecules are hydrophilic. That means they are water loving. So a polar molecule will be attracted to water and will dissolve in it. Nonpolar molecules are repelled by water. We call them hydrophobic. Literally, this term means fear of water, and this means lover of water. And we don't quite use it that way, but they are attracted to water, hydrophilic, and then hydrophobic are repelled. And the repelling is like when you mix the oil and the vinegar together, they will repel from each other and separate. And that's why nonpolar molecules do not dissolve in water. If you were to put butter in water, it would not dissolve in water. Any question about any of that? Hydrogen bonding between water molecules makes water excellent at buffering the temperature. And those of you who live in Western Oregon and Washington know that our temperature is buffered because of the Pacific Ocean, and the water buffers the temperature. Why it buffers the temperature is that when normally energy hits a molecule, it causes the molecule to vibrate and move more rapidly. And that is what we call the temperature, the movement of the molecule. The faster the molecule moves, the higher its temperature. And that does happen to water. However, when energy hits water, besides hitting the molecule and making it move faster, the energy can hit the water molecule and then be absorbed by the hydrogen bond, breaking the hydrogen bond. And so some of the energy that you apply to water is taken by breaking the hydrogen bonds. And that does not elevate the temperature of water. And that's why water is excellent at buffering the temperature. There's only a few substances that I can think of that act like water that do that. Like when we heat oil, for example, almost all of the energy goes into the molecules. So almost all of the energy will be used to raise the temperature of the oil. And that's true for most substances. Water is an exception. And I'm trying to think of something that would be similar. Hydrogen peroxide would work similar, but generally there aren't too many substances that do that. And then when the hydrogen bonds are broken with the water molecule, the molecule will then leave, causing the water to evaporate. And that means that the water molecule having the most energy, or the one with the most highest temperature, is the water molecule which leaves. And that keeps the water relatively cool because it's the hotter water molecule which is leaving. And that is actually important for animals when they are perspiring. We perspire to cool the body temperature, and water is a good substance to do that. And then another good reason is, like I said, the hottest water molecule leaves, and that keeps the rest of the water molecule or the animal a relatively cool temperature. Any questions about any of that? All right. Water can split. And let me put this out because this is one of your questions and a lot of people seem to get it wrong. H2O, I can't right here make a subscript on that too. Water splits into H plus, which I really need the subscript here, plus OH minus. So water molecules can split up, and they do this all the time in liquid water. The charged molecules of water can participate in chemical reactions, and that is really why water can carry electricity. It's because there are a few ions in the liquid water. And if you don't know, salt water is much more conducive of electricity, like ocean water, compared to distilled water. And it's because there's more ions in salt water, the number one being sodium chloride, breaks into ions. The splitting of water into H plus and OH minus also gives rise to the water's pH, and we'll talk about that. Let me state that the water splits from H2O into H plus. This is a proton, a hydrogen ion, a proton. And then OH minus is what we call the hydroxide ion. We'll talk about hydroxide a little bit later. All right, I mentioned that water, when it splits, gives rise to the pH. When we have neutral water, let me back up and delete that. When we have neutral water, whoops, then we have the same number of H plus in the water as we have OH minus. And that is a pH 7. The pH is actually a measure of the hydrogen ions in solution. And for a pH of 7, let me end the show and go ahead and write that. For a pH of 7, when water splits apart, into H plus OH minus, and this time I can do a subscript. At least I think I can. I probably have to make that bigger. So you can see it. Come on. So when water splits apart at pH 7, we have one hydrogen ion. Let me just copy this. I don't know what's going on there. One hydrogen ion in 1 times 10 to the 7th, oops. I should probably put that down there first. Water molecules. Everybody get that? So a pH of 7 means we have one hydrogen ion for every 1 times 10 to the 7th water molecules. A pH of 6 equals 1 hydrogen ion and 10 to the 6 water molecules. And that's how the pH works. Any question? Oops, no, don't need that. No questions? All right. So at a lower pH, you have more hydrogen ions in solution, and then there's more hydrogen ions than there are OH minus. And this we call an acid when the pH is less than 7. When the pH is higher than 7, we call it an alkaline or a base. And there we have more OH minuses into solution than we have H plus in solution. So that a pH of A, well, actually, let's just go ahead and copy the whole sentence and I'll just change it. So for a pH of A, we have one hydrogen ion in, you got it, 10 to the 8th water molecules. Any question about any of that? I'm going to assume everything is good because I can't see your faces and there's no comments. This is showing you a pH scale. The pH scale ranges from 0, where for every water molecule, it becomes a H plus molecule and no H minus. And then lime water, which is a pH of 14. And for every H plus ion, there are 1 times 10 to the 14th water ions. And that's the highest the pH scale can get. Lime water, if you know what that is very close to pH 14. Oven cleaner is close to that as well. And then the strongest thing that you're probably familiar with is stomach acid, which is actually between pH 2 and 1. And then pH 1 will be the car battery acid, a real strong acid. I don't know of anything that's a pH 0. Any question about any of that? Lemon juice is something that, and vinegar is probably the 2 things that you know that are most acidic. And the most basic would be oven cleaner. And household bleach is also very basic. All right, let's talk a little bit about acid, bases, and salts. We talked a little bit about acid and bases. An acid is a substance that disassociates, giving up 1 or more H plus ions in solution. So if we have HCl gas and put that in liquid water, it can get dissolved in the water, and then the HCl can break apart into H plus and chloride minus, and this is what we call hydrochloric acid. Any question about any of that? Now, a base is a substance that your textbook defines that gives off OH minus, like sodium hydroxide can break into sodium ion and hydroxide ion. The hydroxide goes into solution, making the water basic because it's adding OH minus to the water. However, I would like to point out that hydrogen carbonate can break into plus H plus, can make carbonic acid, which will then dissociate out of the water, precipitate out of the water. You get too much of it. The point is that sodium carbonate is a base, and you add it to water, it will make it basic. And it's not because it's adding OH minus to the water, it's because it's absorbing H plus from the water. And so a base really should be defined as a substance that either gives up OH minus or absorbs H plus. Any question about any of that? And yeah, your textbook tutorial for some reason doesn't talk about a base can be a substance that absorbs H plus. Any question about acid and bases? Can somebody please confirm that you can hear me?
Yes, you can hear
I
you.
can hear you.
Yes.
Great. All right, the salt, did we talk about everything? I haven't talked about a salt yet, so let's talk about a salt. A salt is a substance that dissociates into cations. That's a molecule with a positive charge, and anions. That's a molecule with a negative charge. Neither of which are H plus or OH minus, meaning if you were to have sodium bicarbonate, we don't call that a salt because that has hydroxide in it, and that we call a base. The salt is something that disassociates in water and doesn't change the pH, like sodium chloride can dissolve in water and dissociate into sodium ions and chloride ions. And when sodium chloride dissolves in water, it does not change the pH. And that's what we call a salt. And actually, as you know, sodium chloride is commonly called table salt. However, by this definition of a salt, any compound that were to dissociate into a positive cation and a negative anion, we would call a salt, like magnesium chloride is a salt. Any question about any of that? Now, salt dissolve into electrolytes. An electrolyte is just a substance that releases a charged particle into the water. And that could be a cation, an anion, or both. Usually it's both, but it doesn't have to be. An electrolyte is a substance that helps carry electricity. And as I stated before, salt water is better at conducing electricity than distilled water. And the reason is because of the sodium chloride ions dissolved in the salt water. And the sodium chloride are electrolytes. Any question about any of that? All right, organisms need to maintain a fairly constant balance of their cellular acids and their bases to remain healthy. We're not going to talk about this, but in your previous class, this term is known as homeostasis. You don't need to know it for microbiology, but you were taught homeostasis in an earlier class. And how organisms do that is they have buffers in them which help regulate the pH. In our blood, for example, we have hydrogen bicarbonate. No, this is hydrogen carbonate, sorry, or carbonic acid. I can't get it there. I should put that in here first. So we have hydrogen carbonate, also called carbonic acid, in our bloodstream, and it can disassociate into H plus ions. So let me just go ahead and let me just carry that. You can disassociate into hydrogen ion and hydrogen carbonate. And this acts as a buffer. Because it can, oops, that's not what I wanted to do. Because this can put hydrogen ion in the blood, like for example, if you don't have enough hydrogen ion in your blood. And let me continue. And if the blood needs more hydrogen ion, you can break this apart as well. Forming hydrogen ions and carbonate. And this reaction can go the other way if we ever need to remove hydrogen ions from our blood. The reaction can go this way, which I'm just going to change that to this way. Yeah, that doesn't look good. And if still we need to remove more hydrogen ions, this can join with this, forming carbonic acid. Anyways, the point is that this and this and this work to regulate your body's pH, particularly the blood. And I believe these two are the most critical, that this can happen, but that's at extreme alkalinity, is that right? Meaning extreme no, this would be extreme acid conditions. This gets together with that, which is a very low pH, which the blood is not going to be at. All right, any question about any of that? We talked about the pH being the amount of hydrogen ions in solution. It's strictly speaking, the pH is equal to the negative log of the hydrogen ion concentration in solution. And you don't really need to know this formula, but you should know the pH is equal to the amount of hydrogen ion in solution. You should also know the pH scale is between 0 and 14. And that each increase in the pH is a 10-fold increase in the hydrogen ion concentration. Most organisms grow best between a pH of about 6.5 to 7.5. It isn't true for all organisms, but it's true for most organisms. And that would be true for us. The body is about pH 2, and the blood is about pH 7.4. Between, like I said, pH 6.5 and 7.5. I don't know why the blood is slightly basic, but it is. All right, any questions about the pH and acid bases? If not, let's go back to talking about organic chemical compounds. We're seeing some, well, actually all of these molecules here are organic compounds. They can be simple, like methane or ethane, or even acetic acid is pretty simple. Or they can be complex, whatever this is. I can't even read it. A phospholipid. Organic compounds tend to be complex, like these or these or these. They tend to be held together by carbon, like in the acetic acid. All of the atoms are held together by the carbon. So every molecule, except for this hydrogen here, which binds to the oxygen, every molecule other than that binds to the carbon. Carbon holds the molecule together. That's particularly true of larger organic compounds where most of the molecule is held together by carbon. So like in a phospholipid, all of these links here are held together by carbon. It is true that from this carbon to that carbon, we're held together by oxygen. And then by this carbon to the phosphate group, once again, we're held together by oxygen. Of course, there's no carbon here to hold it together, but the point is that carbon tends to be the backbone of an organic chemical molecule. That doesn't mean that other molecules cannot, or other atoms cannot hold the molecule together. Like I stated, right here, oxygen is linking this carbon with that carbon. But there aren't very many of them. Most of them are carbon links, holding the molecule together. Any question about any of that? All right, what's an organic compound? It is a compound that does contain carbon. An organic compound also contains the element of hydrogen. So all organic compounds have carbon and hydrogen. Most have other elements. So they may contain oxygen. Let's go back to this one here. This organic compound had oxygen. This one has oxygen. And I'm gonna blow this up. None of these have oxygen that I see, but let me blow it up so I can see better. So they may contain oxygen. They may contain many other elements like nitrogen, sulfur, phosphorus. All of them tend to be held together covalently by a carbon covalent bond. Compared to inorganic molecules, organic molecules are more complex. Like the common organic molecules are oxygen and water. They're fairly simple, even to acetic acid, which is a really simple organic compound. There's only a few that are more simple than acetic acid, like methane is. Organic compounds are capable of more complicated biological functions. And what's an example of an organic compound? What's an organic compound that you know?
Methane?
Say again?
Methane?
Well, yeah, that was one on the previous slide. I'm trying to say one that you know.
Formaldehyde?
Say again, I'm not hearing you, I'm sorry.
Sorry, formaldehyde? Is that an organic
Yeah,
one? or is
that's an organic compound.
that- gotcha.
Any of the biological molecules, and practically any molecule containing carbon and hydrogen, will be an organic compound. And they are more complex than the inorganic compound. And some inorganic compounds that we'll talk about are oxygen and carbon dioxide, as well as water. Amino acids, carbohydrates, nucleotides, lipids are all various organic compounds. Let me come back to this slide here. All of these molecules are hydrocarbons. A hydrocarbon is an organic compound, but they're unique in that a hydrocarbon has very few, if any, molecules other than carbon and hydrogen. So that's why it's called a hydrocarbon, because it's composed of hydrogen and carbon, mostly. Like I think there might be one oxygen molecule in the gas you buy in your car. And all the rest of the molecules are carbon and hydrogen. Methane, for example. Let's go back to this slide. Methane and ethane gas would be a hydrocarbon. Organic compounds tend to be held together by carbon. Carbon is important because carbon can bind with up to 4 other atoms. Like in methane, shown here, carbon is binding to 4 hydrogens. In ethane, carbon is binding to 3 hydrogens. And this carbon is binding to 3 hydrogens. And then these 2 carbons, their 4th bond is to each other. The carbon can be in a string structure shown here, and a better example would be here, or it can be in ring structure shown here. Talk about that. Oh, on the carbon skeleton backbone, which is the carbons holding the molecule together in a covalent chemical bond. So right there, it's a covalent chemical bond right there. You can put functional groups where a hydrogen would be in a hydrocarbon. And then the functional group can influence the chemical behavior of the organic compound. why organic compounds have the properties they do have is because of the functional groups attached to the organic compound. So functional groups are responsible for the chemical and physical properties of a particular organic compound. For example, the hydroxyl group, OH, is found in alcohols, and it makes the alcohol hydrophilic so that it's attracted to water molecules. And then the OH group also helps the methanol dissolve in water, making alcohol. Let me talk a little bit about that from this slide here. When we're talking about methanol, we have methane shown here. And one of the hydrogens is replaced by an OH group that changes the properties of this molecule. Methane is hydrophobic, does not like to dissolve in water. And then you add the OH group, just take off the H group, or an H. You then change the properties of methane, and we call it methanol. And instead of being hydrophobic, it's now hydrophilic, meaning methanol will dissolve in water. Any question about any of that? So the functional group changes the hydrophobic molecule to a hydrophilic molecule, even though you're only replacing one of the 4 hydrogens. You do need to know the functional groups shown in red. We've talked about OH groups found in alcohol, carbohydrates, proteins, lipids. You do need to know the amino group, and that is an NH2 or an NH3 group. Amino groups are primarily found in proteins or amino acids. And this amino group does bind to a carbon, but the carbon isn't part of the amino group. And then the R stands for the rest of the biological molecule, so that R is just the remainder of the molecule and can be essentially anything. You do need to know the carboxyl group, which is CO with a double bond and then OH. And you may see that written out instead of like that. You may see it linear, like this. Or you can see it like that. Oftentimes we do write it, especially just in text, we write it like that. Write it in superscript. You should also know the phosphate group, and that is PO4, meaning 4 oxygen groups, and it has 2 charges, one complete negative charge there and one complete negative charge there. You can find phosphate groups in phosphate. You can find it in DNA and RNA and in ATP. I didn't mention, but carboxyl groups can be found in organic acids, lipids, and they're particularly found in proteins. So here we have an amino group, and it has a central carbon. It has an amine group, the amino group on this side, and a carboxyl group on this side. There's the carboxyl. And this is actually amino acid. We'll talk about that in just a few minutes. Any question about the functional groups? Well, if there's no questions, let me ask a question for you. Why am I asking you to know or memorize the functional groups labeled in R, in red? Why am I asking you? Come on.
Well, the phosphate group, it's clear that it's an important functional group in ATP and DNA.
That's true. What about the other groups?
I'm guessing we could potentially find those in bacterial colonies or in maybe infectious microbes or something.
You can find these functional groups in other biological molecules. So yes, you do find them in bacteria and other microorganisms, but it's more than that. You find them in everything that is alive or was once alive. And because of that, I want you to know these functional groups because they're a little more important, at least to biological molecules. Any question about any of that? All right. The small organic molecules can combine into larger organic molecules that we call macromolecules. Macromolecules are polymers consisting of many small repeating molecules, and these small repeating molecules are called monomers. Any question about any of that? So when we're talking about the biological molecules, they can be a macromolecule like a protein, or they can be a small molecule, which can actually make up the protein. And if we're talking about protein, the small molecule, which is a monomer, would be called an amino acid. Any question about any of that? All right. Organic chemical compounds can be made larger or just made by dehydration synthesis, where we can connect one monomer here to another molecule, which may be a monomer, or it could be a macromolecule minus the one monomer to make it the polymer. But anyways, the point is you can combine a monomer with either a monomer or another molecule in condensation reaction, which is also called dehydration synthesis, where you then break off the OH group here and the H group there, which will go on to form water. And then you link this R directly to that R in dehydration synthesis. And that is how our body makes macromolecules that we call polymers from the monomers. Like when you're building a protein, you join two amino acids together, which both would be monomers, and then you get the dimer. And then you take the dimer and add another monomer, and you get a trimer, meaning three, and then you keep building that way. So dehydration synthesis is a mechanism for synthesizing macromolecules such as carbohydrates, lipids, and proteins. And this is showing you dehydration synthesis, where this monomer gets together with a molecule which is very large, but it may not be a polymer yet, or it may be. And then the monomer binds by forming water. The hydrogen comes off of this molecule. The OH group comes off of this monomer. The monomer is directly connected to the carbon of the polymer. And then the H and the OH combine to form water. Hydrolysis is just the worst. Reverse, where water comes in, breaks this bond between the monomer and the polymer. The water breaks apart, the hydrogen going here, the OH going, attaching to the monomer. And then this monomer can leave the reaction. Any question about any of that? No questions, let's move on. Let's talk about the first of the biological molecules. These are carbohydrates. They include such things as sugar, starch, and other complex carbohydrates. Like you can find complex carbohydrates in bread, or even in bread flour. Carbohydrates are an important energy source for animals. They are important for the structure, and they're important in the nucleic acids of prokaryotes. And this is true actually for all living life forms, where carbohydrates are the primary energy source. And then they're important in building our body structure. We can have carbohydrates on the outside of your cell. And carbohydrates are important in the building of nucleic acids. We'll talk a little bit about that later in the lesson. Carbohydrates are composed of carbon, hydrogen, and oxygen. However, they are composed with a general formula, where for each one carbon, we have 2 hydrogens and 1 oxygen. And then this cycle of carbon atoms and hydrogen atoms and oxygen atoms will repeat in other regions of the territory the carbohydrate is in. Any question about any of that? For carbohydrates, besides having the general formula CH2O, you need to put, for larger molecules, you can still use this, but then you put a large number in for N, and N is just saying the numbers. For glucose, the N is 6, meaning there's 6 carbons in glucose. 12 hydrogen, 2 times 6 is 12. And then there's 6 oxygen in glucose. Any question about any of that? Carbohydrates may be simple sugars or strings of simple sugars linked together. And if you link enough of them together, you will no longer have a simple sugar or a dimer. You will have a large, very large organism composed of the molecules bound to carbon. And what am I trying to say here? Sorry, I'm drawing a blank here. If anyone knows what I'm trying to say, you can state it and give me a hand here. Let me go back. A carbohydrate is composed of sugars or strings of sugars, like sucrose, which is a dimer, is composed of 2 monomers cooked together. However, starch is composed of glucose, but this glucose is repeating many, many times. And they're just all hooked together in starch. We can classify carbohydrates into three major groups. The monosaccharides, these are simple sugars. The disaccharides, the table sugar is an example. Another name for it is sucrose, the disaccharide. And then polysaccharides. Poly means many, saccharides. Saccharides means sugar or glucose. So a polysaccharide means many sugars. Sugar molecules, I guess I should say. Any question about any of that? Monosaccharides are simple sugars. They are always between 3 and 7 carbon atoms, making up the sugar. Usually, a simple sugar or a monosaccharide is composed of 5 or 6 carbons. Like glucose has 6 carbons, fructose has 6 carbons, ribose has 5 carbons. But there are a few exceptions, and we're not going to talk about the exceptions. And that's why we say that this monosaccharide is between 3 and 7 carbon atoms. The disaccharide is above this number. It's the joining of 2 monosaccharides together, such as when we make sucrose, table sugar, from, oops, too far. We make table sugar, sucrose, from 1 glucose molecule and 1 fructose molecule. And they, of course, form by dehydration synthesis. The water comes out, and then this carbon binds to the oxygen on this, the structure. That we call dehydration synthesis, synthesis to make a bigger molecule. And we can go in the reverse with hydrolysis, split this bond right here with water, and then the water separates going to both of the monosaccharides. Polysaccharides consist of many monosaccharides or many sugars. The polysaccharide may be straight or branched. Examples are shown in this picture. Starch, which isn't actually shown, although we have starchy bread and starchy rice and starchy potato, as well as beans and chestnuts. Most of you probably don't know, but beans and chestnuts are a good source of protein, but only about 1/3 of the weight is protein. Another third is lipids, and then the other third would be carbohydrates. So beans and chestnuts and other nuts are actually good at supplying hydrocarbons. Any question about any of that? We will talk a little bit about starch, one polysaccharide. Very slight discussion of glycogen, and we won't talk about the others. We do talk a little bit about cellulose. All right, let me see if I can blow this up because I'm not seeing it. So we're looking at a table of polysaccharides. You do not need to memorize this table. So do not memorize it. What I'm just trying to show is for both starch and glycogen, they're held together by, their glucose held together by a single covalent bond. However, there is a difference between starch and glycogen. Starch is the way that most plant cells store carbohydrates. And that's why we can get potato starch and corn starch. But their seeds do contain other molecules. Let me state that although glycogen and starch are the same, sugar molecules linked together, one after the other, the chains are different. For the most part, starch is a non-branching chain. And then glycogen is a branching chain. Cellulose we'll talk about in just a few minutes, I think. But it's another of the biological molecules, but no, it's another example of a polymer. Any questions about any of that? All right. If there's no questions about carbohydrates, I think I will discuss this slide and then end this lesson because we're ending at 6:20 and we're getting close to that time. Let's talk about the second of the biological molecules. These are lipids. And when we're talking about a fat, that is a term for a lipid. However, lipids is more than just a fat because long chains of hydrocarbons are a lipid, but they are not a fat. And then if you go out into the world, like you can find oil, it's a polymer of, well, I'm not sure what oil is a polymer of. It's an organic compound, that I know. And it was once alive, although that was a long, long time ago. And then the Earth's geology converts it into oil. The point is, oil is a lipid, and lipids consist of carbon, hydrogen, and oxygen. And they do have oxygen. However, they do not have very much oxygen. And let me just show you a lipid right here. The only oxygen is right here, and then right here, but this comes off when this fatty acid tail binds to whatever it's binding to, like glycerol. So that OH actually comes off, leaving only one O, oxygen, in all of this group of a lipid. Any question there, or did I confuse people? Lipids are important in the structural and energy storage roles, meaning you look at your belly, there's a structural layer of lipids, fat, and that's partly to insulate our bodies. And it is true that we can use that like when we're in famine. But in today's day and age, it actually becomes a nuisance. And if you gain too much fat, it'll be unhealthy. Lipids are, for the most part, nonpolar and insoluble in water. There is one exception, and we'll talk about that next time. The exception, I'll mention what it is, but I'm not going to discuss it further. And that is a drawing a blank on what that's called.
Is it a lipoprotein?
Phospholipid. No, it's a phospholipid. That is one lipid which actually is soluble in water. And that lipid is not hydrophobic, at least not entirely hydrophobic. And then lipids are a primary component of cell membranes. Cell membranes, the main molecule is a lipid, although it is a phospholipid molecule. All right, lipids can be broken into 3 different groups: simple lipids, complex lipids, and steroids. Are there any questions? If not, I will see you in the lab at 6:30. So I'm going to say goodbye. Bye.